All of our programs are allogeneic. They're all off the shelf. They're in vials. They're cryopreserved. They can be thawed and shot into patients' arms at the site of therapy, so outside of a hospital-based setting, potentially in a community-based setting. We start with a natural killer cell. We start with a donor-derived cell. We use proprietary expansion technology from our founder, Dario Campagna, K562 cell line. We rapidly and robustly expand these cells over the course of 14 days of manufacturing, not 45. And we believe we can have a very attractive cost of manufacturing. At the moment, it's about $2,000 a dose when we look at producing maybe better than 500 doses per healthy human donor at full capacity. And this is a very advantageous cost per dose and will lead to a very attractive cost of goods and allow us to use these cells to not just dose a patient, but to dose a patient multiple times, multiple doses, multiple cycles, multiple potential lymphodepletions or not. And the ability to dose these like an antibody or like a chemotherapeutic, these cells tend to go in, do their business, and go out, shrink the tumor, and have the kinetics of traditional therapy. So very amenable to multiple dosing, very amenable to off-the-shelf delivery to the patient at the patient's bedside. And as I mentioned, in an outpatient setting, and without the toxicity that has been associated with the currently available autologous CAR T cells, no appreciable neurotoxicity, graft versus host disease, or cytokine release syndrome. We have two co-lead programs. The natural receptor on the NK cell is NKG2D. We have an NKG2D engineered program, as well as a CD19 program, NKX019. NKG2D, or NKX101, is in phase 1, dose escalating at the moment. That IND was cleared in July of last year, and we just recently had a clearance of our CD19, NKX019 IND. So the 101 trial is enrolling patients in dose escalation, and the 019 program will be starting to enroll patients in the second half of this year. So 2 INDs in the course of a year after we went public. Our platform is strongly differentiated. As I mentioned before, we start and finish with this natural killer cell. We expanded over 14 days. We're not differentiating from a stem cell. We're starting with a natural cell in high volume. And so we can expand them in high volume, and the manufacturing is rapid and prolific. So these cells don't have to go through rounds of manipulation. They get expanded, they get manufactured, and they're ready to go. And we can cryopreserve them, as I mentioned before, put them in vials, and then thaw and shoot these cells. At the end of the day, the way we look at the NK cell space is that what matters is what these cells are going to show clinically. The starting material may not be an issue down the road. And we are in the clinic now and looking forward to presenting data for 101 at the end of this year, a handful of patients, and CD19 data next year sometime.
Excellent. Great overview and setting the foundation for the discussion. So some of this will be kind of integrating a lot of the discussion points investors have around the emerging space of NK cell adoptive therapy. Maybe one place to start, though, and we hear this a lot, and it's always, I think, hard for investors to really understand what the truth is behind this. But talking about the supply of source NK cells, how you approach this issue in the way you've gone about the manufacturing and the donor selection and source versus others that might use induced pluripotent stem cells as starting source. Every company says they do it better and have some advantage. Have we seen any proof in that? Have we seen any reason to actually believe that one cell source is better than another cell source?
Absolutely not. So when people don't have a lot of clinical data to share, they talk about starting material. We're happy to talk about it too, by the way. The reason we like what we're doing, and by the way, we like all the other sources as well, and we've looked at them and we've made determinations that we're going to go with a healthy donor-derived product, mostly because we just felt that we could expand them, proliferate them, and produce them much more quickly and with much less variability. So that's why we went with this platform. It doesn't mean that we may not at some point go to iPSCs or cord-derived cells. And again, we are of the firm belief that what really is important is, at the end of the day, clinical data. So whatever your platform is, whatever your starting material is probably going to become irrelevant when there's ample clinical data to talk about. And we think we're all going to be generating that over the course of the next couple of years. But one of the reasons we like healthy donors, I mentioned before, is we can take Dario Campagna's K562 cell line, and over the course of 14 days, by adding membrane 15, by doing the other bits of technology that we've added to our proprietary technologies, we can create over 14 days, 500 plus doses per healthy human donor. We can select healthy donors for characteristics, like, for example, if we want ADCC and CD16, we can select for someone a donor that has this CD16 characteristic. So we can do all the things you could do with other forms of engineering with just starting with a donor-derived cell, and then you can expand them rapidly, make them quickly, make them in great numbers, and make real NK cells out of NK cells. So we think it provides us with a healthy NK cell starting material. Does it mean it's better or worse than other starting materials? No, it means that we have great starting material, and we're going to make cells, we're going to put them in the clinic, and I'm going to talk to you about what that looks like. NK cells in general are highly cytotoxic. They have the ability to select for desired features in our donor pool, as I mentioned before, and we can take this rich source of cells, expand them, and come up with a cost of manufacturing that at the moment looks to be about $2,000 a dose, and that enables you to dose it multiple times, to dose multiple doses in a cycle, to dose multiple cycles, and to give you a lot of flexibility.
So as your team's ramping up on the clinical side, obviously you're thinking about building the manufacturing capacity out to support late-term clinical studies, pivotal studies, and then bridging into commercial GMP in-house manufacturing. What are some of the milestones that your team's focused on over the next, call it 12 months to continue to ramp that up in parallel with the clinical studies and the demands of enrolling patients and treating more patients with more doses?
Right, Ralph, why don't you talk about how we're doing with our clinical GMP facility and then with our commercial GMP planning?
Yes, absolutely. So we built out our clinical GMP facility and brought it online. This facility is designed to support the early clinical development pipeline. And the first product that we will make there is for the NK019 clinical trial. All future programs will be manufactured there for early development, and we'll eventually also move NK101 into this as well. In parallel, so this facility is up and running. In parallel, we're in the design and engineering phase of a second larger facility. We expect to move forward to this facility with construction in 2022 and start planning activities, qualification activities for this in 2023. That is going to be a large-scale facility where we have planning to have 500 plus doses per batch, and that capacity of that facility is anticipated to be sufficient to supply people to clinical trials, for our lead programs, and then also for commercial launch.
Excellent. So as your team builds out the clinical efforts, you're obviously looking at both hematological and solid tumor indications, very different approaches and outcomes to date and state of any cellular therapy that's been tried. From your team's perspective in terms of developing that strategy of how to design clinical studies and where to go in terms of different tumor types, what do you think we've been able to achieve to date in the hematological and solid tumor side? And what are kind of the learnings that you've applied to thinking about bringing some of your programs forward that might be able to get around the hurdles that your predecessors have faced?
Sure. Maybe, Nadir, you can talk a little bit about the platform and the CRISPR deal and how that enables us in solid tumors as well as heme malignancies. And then, Kanya, you can talk about how we chose these indications and what we expect to see.
Yeah, I mean, I'd say as you look at the space, obviously between the autologous CAR Ts and where a lot of the other modalities that are trying to move into the more allogeneic space are starting, everybody's kind of starting in the hematological setting. I think sort of lower hanging fruit, accessibility, proven concepts there with the autologous modalities, I think creates sort of a good entry point. As we have thought about sort of the evolution of the platform, and as we move from our first couple of programs, which we're also starting in heme, into the solid tumor setting, we firmly believe that NK cells engineered are going to be superior than a non-engineered NK cell. And the question is, what additional engineering steps beyond having a chimeric antigen receptor and cytokine support, as we have in the memory-bound IL-15, can further enhance the platform and the cell product to give the best shot in a solid tumor. And so gene editing is something that we've looked at, and we recently announced a partnership with CRISPR Therapeutics, whereby we're going to work collaboratively to co-develop, co-commercialize a couple of different programs. The first is targeted against CD70 expressing tumors. This will be a CAR NK that's going to be gene edited. That's going to go into solid tumor settings as well as potentially some heme settings. And then we've also got a really exciting program where we're combining the allogeneic NKs again with the allogeneic CAR Ts, kind of bringing the two worlds together in a single product with the power of both the innate and adaptive immune systems. But in addition to those collaborative products, we have now access for our own platform, the ability to incorporate gene editing through a license that we get from CRISPR. And so that allows us to now kind of take our platform and the next set of our product candidates into the next generation that include gene editing components as well, which I think will be particularly important as we foray into the solid tumor setting.
Great. And maybe, Kanya, you want to talk about how we chose AML, MDS, and then the solid tumor for 101 as well as CD19.
Yeah, happy to. So for NKX101, we looked at a couple of different things, mainly the system. There's a wealth of clinical experience with non-engineered NK cells. Nearly 600 patients have received this product. Non-engineered NK cells. It's a single institution study. Vast majority of them patients with AML. So as shown here on the slide on the right, that from that data, we know that patients with relapsed refractory AML in single institution studies with a single dose of non-engineered NK cells have seen responses between 20 to 50%. So that's one of the reasons we picked relapsed refractory AML or MDS patients as our first in human trial. Now, if I could point your attention to the table on the left, that shows the vast variety of tumors in which the NKG2D ligands have been well documented to be upregulated. So in other words, NKX101, which is the NKG2D receptor found naturally on an NK cell, recognizes 8 known ligands that have been well documented across these variety of tumor types. So that's how we think of further tumor indications as well. So we do have plans to evaluate NKX101 in solid tumors, for example. And so really, we are building off of existing data as we think of the indications and how we are planning our clinical studies.
Excellent. And when you're thinking about maybe sticking on this slide, which is really interesting, in AML specifically, how do you think about the right line of therapy to explore and where you would eventually want to go over time given you have very distinct patients, that upfront can or cannot tolerate intensive chemotherapy, and there's still unmet need across actually both patient populations, but one would eventually go to transplant. One might, if they're lucky, get a mini transplant. So yeah, how are you thinking about the clinical design in AML specifically over time?
So our current ongoing trial enrolls patients who have relapsed refractory AML. In other words, they failed current available therapy. They should have failed at least one before they come on our trial. So the way we are thinking of our AML development plan is, as data supports it, this could potentially be further expansion cohorts to look at a pathway towards registration for this really high unmet need patient population, where the median survival is about 3 to 4 months. And along with that, we are also thinking of moving our product into earlier lines of AML treatment upfront, either with the low-dose intensive agents, either hypomethylating agent and our chemotherapy for the younger fit patients. So it's our intention to have a comprehensive AML development plan with our product, assuming, of course, our clinical data supports that.
And would you consider, so I guess maybe thinking about how the treatment landscape might be evolving a little bit, and thinking about the recent approval of Onureg, which is actually being used in the maintenance setting. How soon would your team start thinking about potential combination therapy with an NK adoptive cell therapy, which I think a lot of investigators would be interested in?
Okay.
So the way we are thinking of our program is, as we are still in dose finding now, as we achieve our target dose, that's when we are thinking of layering in combinations as well in an earlier line of indication. Because our approach here is to develop this product. Our clinical development plan is focused on making this, on obtaining data to support registration. So in this late line patient population, there are no other real approved agents for use. So our intention is to go combination earlier as we identify our dose and go into expansion cohorts.
So Dane, just to put a point on this, we really do believe that it's important to know what your drug or your cell does as a single agent first. So we're steadfast in doing the single agent dose escalation, and then we'll expand after that, as Kanya just said. So once we know, and even if we go to a high dose and we can go higher, we might even go higher. We might go higher at the same time, expand into a combination the way you would in an IO expansion in a phase 1 kind of trial situation.
And that's a great bridge into just setting some expectations and any details you can provide of what first data might look like from the phase 1 dose finding study. What your team will be focused on in looking at that initial data from a safety and efficacy perspective, and what kind of reasonable assumptions are given the patient population of what would constitute a meaningful clinical effect. Yeah,
Kanya can speak to a meaningful clinical effect. It's better than what's available today, standard of care. Number one, there isn't a lot available for these kinds of relapsing refractory patients. But our goal is to do better than standard of care, and our goal, we'd love to do as good, if not better, than what we're seeing now in these early stage studies, but perhaps with more dose escalation and a dose response, perhaps. That's kind of what and again, we're guiding to a handful of patients by the end of the year. We're going to stick to that guidance, and we hope to have the patients and the data to show people by the end of this year. Tanya, anything else?
Yeah, so what I would perhaps add is that in this patient population, as Paul said, there really is no standard of care for relapsed refractory AML patients who have various mutations like IDH1, IDH2, or FLT3. There are mutationally selected agents for a subset of population. Broadly, apart from gemtuzumab, which is approved for these patients as monotherapy use, which is seldom used because of its toxicity, especially when HCT is part of the overall treatment plan. There really is very little for these patients, maybe recycled chemotherapy for those who are fit enough to receive it. So our thinking is that a response rate of about 25-30% would be very clinically meaningful for this patient population, which lasts for about 4 months. I mean, as a further example, flotetuzumab, which is a DART, which has gotten some attention, they had an 18% CR rate in a fairly similar patient population. So that's one frame of reference as we think about how our patient population with a median survival of about 3 to 4 months and coming in after having exhausted all other therapies.
So you would think that the patient population could look like some of those initial studies, like with flotetuzumab?
Right, we are targeting a relapse refractory.
Yeah. Okay, great. That's super helpful. Maybe transitioning now to the NKX019 program. Let's start with the timeline that you think could be first data from that program, given how the entire entirety of programs focused on lymphoma have been progressing recently. I know you're still in the IND, seeing the IND open on that. But generally speaking, let me frame it this way. Given the increasing complexity of the lymphoma space, how are you thinking about that first clinical study design that's going to get you the answers you want to then figure out how to further progress the program?
So with the CD19 trial, the current available CD19 therapies, there's really two major issues which affect their broad usage, right? And our NKX019, we believe, is well positioned to address both of those aspects. One is the safety profile. With the T-cell based therapy, with its increased expansion and so on, there's a lot of cytokine and an immune system run amok kind of side effects, CRS, ICANS, what have you. With an NK cell, where the intrinsic biology doesn't lend itself to that sort of a massive expansion, we expect the safety profile to be much more suited to outpatient administration, even in a community setting. Now, the second aspect, of course, with autologous cell products is the weight, and here being completely off the shelf and allogeneic, it addresses that as well. I think that's really how we see the unmet need for NKX019 is in addressing both the huge efficacy, the safety aspect, as well as the quick availability and off the shelf with our manufacturing capabilities.
So, and Dane, that IND has been cleared, and we're going to dose a patient in the second half of this year. But our strategy is to go beyond the U.S. and the investigators we're talking to lack no enthusiasm for another CD19 CAR-NK program, one that's allogeneic and off-the-shelf, cryopreserved in vials. So we're feeling really good about it. We'll see when we start enrolling, and then we can provide some further guides in terms of how fast it enrolls.
So let me ask this question. As you think about the program, there's an ongoing debate for an off-the-shelf cell therapy, whether it's a T-cell or NK cell in lymphoma. Is the appropriate comparison to auto CAR-T, or is it more appropriate to compare these therapies to infusion-based products like the emerging CD20, CD3 class?
Kanya, maybe you and James want to talk a little bit about mechanism and where we think the market may be going.
Yeah, so at the end of the day, Dane, you're right. For the patient, it's not so much about what makes the product, but what it can do to the disease. So that would be, whatever is the standard of care is the appropriate comparison. And so when we think of our attributes of NKX019, retaining efficacy with that safety profile, that outpatient use, we are thinking of it broadly in the context of what's available in the landscape for those patients and not just on autologous CAR-T.
And that kind of bridges into the clinical effect of that debate. It really comes down to, okay, for these off-the-shelf cell therapies, should we have the expectation that X number of patients should be getting long-term durable complete responses to the magnitude that we have seen with auto CAR-T in lymphoma? Because that process, to a certain degree, is almost more like a transplant process than, even though you're using lymphodepletion, than you would be going through if you're getting NKX019. I know it's a highly philosophical debate right now, but it's pertinent to the clinical community in terms of how these therapies get developed potentially into earlier lines.
So, you call it a philosophical debate, but I think that's really a key question. And the whole field is learning, right? Can the allogeneic cell therapies recapitulate the durable response of autologous cells while bringing in some of these other advantages that I spoke about? So I can talk to you about our approach to how we hope to achieve that. So what we are planning on doing, as laid out here very nicely in the slide, is we give lymphodepletion, and then we give three full doses of cells within the 2 to 3 week window where the host immune system is suppressed. So then we do an assessment day 28, depending on if at least there's a partial response or some sort of a reduction in blast, depending on the tumor type, and safety profile permits it to give additional cycles. So we think of it as achieving a deep remission to then translate into durability of response. We know that our product, being allogeneic, is going to eventually be cleared by the host immune system. So our entire focus of our clinical plan is to achieve that depth of response as quickly as possible.
And if you can do that, if you can get the same response you get with an autologous CAR-T with an off-the-shelf NK cell, I can tell you from the very early days when we started the company and started talking to partners, we spoke to all the CD19 autologous CAR-T players, and they were all very interested in an NK cell off the shelf that would do the same thing, but in a much safer way, without the toxicities and with the ease of an outpatient administration. So don't think there's any question that if you were to do just what they do, but with an NK cell, you'd have a very nice product at the end of the day.
Excellent. And in a few minutes I have remaining here with your team, I did want to touch on your first solid tumor effort, which is going to be CD70 targeted. Some of your peers are in the clinic now testing a CD70 cell therapy. This is also your first collaboration that will be clinical, I presume, with CRISPR Therapeutics. What does your partnership kind of bring to the table that you think may create a better mousetrap in terms of targeting CD70 than some of your peers? And what's your thought in terms of the overall hurdle of seeing both safety and efficacy with these CD70 approaches?
Nadir will answer that, but before he does, let me just make it clear that our first solid tumor program is NKG2D in hepatocellular colorectal cancer that's isolated to the liver, as well as cholangiocarcinoma. That will be later down the development path of 101 and AML and MDS. And then CD70 will come later. But I think this question is more about CD70 and CRISPR, so let's let Nadir take the last minute and a half and tell you a little bit about that.
Yeah. So I think the beauty of this really is, there's a lot of companies now incorporating gene editing into their cell therapies. When we sort of recognized that this was something we wanted to incorporate, we looked across the landscape of different technologies to edit the cells. and then the partners and players, and what it would really take for us to do this. And where we landed was we really liked the CRISPR-Cas9 system, CRISPR-Cas in general. But then it became a point of, when we did diligence, identifying the partner that had really operationalized the technology. Many people say they can take it from the bench and you can manufacture and you can incorporate it as cells, all this stuff. But really, when you look at the history of how long companies have been working on this, very few have successfully done that and put product candidates in the clinic. And so that's why we picked CRISPR. We couldn't be happier with partnering with them on this. And then I think, as we look at CD70, we know that that's got to be edited fundamentally to knock down CD70 expression itself. Otherwise, you're going to get fracture site, having the cells express the antigen and a CAR. But then there might be other edits we're looking at in the NK cell to enhance some of the NK cell features themselves to give us a even better shot, particularly as you go into the solid tumor space and the tumor microenvironment is going to throw out lots of challenges that the NK cell is going to face. Editing is another tool in our arsenal to overcome those challenges and give us a much better shot and probability of success. So, that's really how we think about it. I think our peers have different cell chassis that they're looking at. But again, I think the NK cell with a CAR, you always remember that the NK cell will still have its native expression of its activating receptors. So it's this multivalency and ability to recognize multiple antigens in addition to whatever the CAR is targeting. That will always be a feature and benefit of the NK approach. So we're excited about this program.
Excellent. Cool. I know I've tortured you guys with a number of questions in terms of the development and evolution of the pipeline, but it's been really great. Obviously, a fascinating space with a lot of opportunity and hope for patients and clinicians to advance therapies in high unmet need. So thank you for doing the work to try and advance these therapies and technology. And thank you for joining me for this fireside chat. Obviously, I think a lot of the audience members probably know you guys already, but if you don't, reach out to Greg. If you can't get a hold of Greg, I'll be happy to link you up with the Nkarta team. But thank you, Paul. Thank you, Greg. Thank you to the entire team for this discussion today.
Thanks for having us, Dave. We appreciate it.
Thank you, everyone. This concludes the session, and